Okay, now we're in the review session, guys. Magic. So talk to me about the exam. Thoughts, concerns, questions? What do we need to know? Yes? Okay. All right. So, yes. So her question had to do with the transport system kicking protons out. Okay. So what was it that was kicking protons out? All right. So if we go back to that, and we talk about the transport system, and you're talking about this figure right here, right? Okay. So in this particular case, the kicking out of the protons is done by a pump, and I didn't say what caused that, but I'll tell you briefly here what can cause that, and you're not responsible for knowing what caused it. All right? Some systems can use ATP energy to kick protons out. Some systems can use light. So photosynthetic bacteria can use the energy of light. Remember I talked about the kicking mechanism? And what the kicking mechanism is doing is it's kicking protons. So it depends on the bacterium in terms of what the actual energy source is that makes that happen. Okay? I promise I will tell you about a photosynthetic fish because the kicking mechanism is essential for a photosynthetic fish. Yes? Well, I will talk about what I talked about with the secondary transport system. So the secondary transport system, the reason I show you the transport system is because it's a really good example of a transport system that's using a gradient to do something. So a gradient is something that's a concentration difference. You can work with a gradient or you can work against a gradient, right? Whether you ski uphill or you ski downhill, right? So if you're working against a concentration gradient, you're moving something from a low concentration to a higher concentration, and that requires energy. If you're working with a concentration gradient, that goes from a high concentration to a low concentration, and that actually is a source of energy. So to answer your question, the reason I show this figure is it shows how a cell can use a concentration gradient as a source of energy. Now the source of energy, my shoe's untied, the source of energy is important because this mechanism is used to bring something against a concentration gradient. Meaning, lactose is in higher concentration in the cell than out of the cell. This concentration gradient is working to bring lactose in. So a secondary system uses a concentration gradient as a way of making something happen that would otherwise not be energetically favorable. Yes? Yeah, good question. Why does a cell even care? Why does a cell want to do this? This is a bacterial cell, and lactose is a sugar that's a source of energy. So it wants that lactose so it can break it down and get the energy out of the lactose. Yep. Yep. Yes? Excuse me. That's a good question, actually. Her question is, can this only happen when there is lactose present? And for this particular transporter, that's correct. Yes. In general, when you've got a system that is specific for something, like this is specific for protons and lactose, it will only work if both things are present. So this isn't just a hole in the wall that something's falling through, but it's actually specific for the molecules that are there. Is this where we apply the lactose repressor? No. However, the lactose that comes in here is metabolized using proteins from the lactose repressor. So it's related, yes. So it's the same lactose, right? But this isn't the DNA system. This is the lactose that's actually being broken down. Okay? Yes, I'm sorry. Yes. Yes?
Just types, two different types of subcategories.
Sure. Okay, so her question is phosphatidic acid and phosphatides. Basically, I think her question is, how does that relate to the other things that I've talked about there? Is that safe to say? Yeah. Okay. So phosphatidic acid is an example of a molecule that we categorize as a glycerophospholipid. And a glycerophospholipid, I said I would also call a phosphoglyceride, right? The relationship between phosphatidic acid and a phosphatide, which was your specific question, is that if I take phosphatidic acid and I join it to something else, I create a phosphatide. So if I take phosphatidic acid and I join the phosphate to a serine, I make phosphatidyl serine. If I join it to choline, I make phosphatidyl choline. Yes? What would I use phosphatidyl? Very good question. What would I use phosphatidyl serine for? It would be a component of my membrane. All these phosphatidyl compounds have a polar end and a nonpolar end. So when we talk about the lipid bilayer, the polar ends stick out facing water, and the nonpolar ends are on the inside. So any of these things, phosphatidyl serine, phosphatidyl choline, phosphatidyl ethanolamine, sphingolipids, all of those would be used to make the lipid bilayer. Okay? Yes, ma'am. Sure. Can I explain the role of sigma factors in the process of RNA transcription? That was your question, right? Okay. So if we think about what has to happen during transcription, first of all, transcription happens at a promoter. Promoter is a specific sequence that's recognized by the RNA polymerase in an E. coli, in a prokaryotic cell. It's a specific sequence that's recognized by the polymerase. The part of the polymerase that does the recognizing of the promoter is the sigma factor. So the sigma factor is the part of the polymerase that recognizes and binds to the promoter. Yes, ma'am. So does the sigma factor have a certain affinity for it? And the answer is it does. I mentioned briefly in class, and I hope it wasn't confusing, unlike everything else, that there are different sigma factors that recognize different promoters, so they'll be specific for certain promoters. If you know that the sigma factor recognizes the promoter, you know enough. Yeah. Yes? What's that? Sure. Okay. So the enzyme ATCase, I'll remind you, is the enzyme that catalyzed that first step in the process of making pyrimidines. I said there were about 10 steps, and ATCase was catalyzing that first step. Right? ATCase was an example of an enzyme that, yes? Pyrimidines. Pyrimidines, yeah. Okay. ATCase is the first example I've talked about in class of an enzyme that's regulated by what we call feedback inhibition. Feedback inhibition. Feedback inhibition occurs when the end product of a pathway binds to an enzyme at the beginning of the pathway and shuts it off. Or I shouldn't say it actually does shut it off, yes. That's the inhibition part. Okay? So that's what feedback inhibition is. All right. Now, her question had to do with the structure of that enzyme. And I said that the enzyme had two categories of, or two classes of subunits. The two classes are called regulatory subunits and catalytic subunits. And it has 6 of each. The catalytic subunits are where the reaction is catalyzed. Remember, it's an enzyme, so it's going to catalyze reaction. The regulatory subunits bind to nucleotides, and those nucleotides change the structure and the activity of the enzyme. The nucleotides binding to the regulatory subunits change the structure and activity of the enzyme. The nucleotide ATP binds to the regulatory subunits and locks the enzyme in the R state. The R state meaning the more active state. So binding of ATP to the regulatory subunits causes the enzyme to be active. Conversely, the nucleotide CTP can also bind to the regulatory subunits, but it will freeze the enzyme in the T state, the less active state. So CTP will tend to turn the enzyme off. CTP is the end product of the pathway. It's feeding back, it's binding to the protein, and it's turning the protein off. Now, there's one other molecule that I described that affects the enzyme, and it actually binds to the catalytic subunit because the enzyme acts on it, and that is aspartate. Aspartate binds to the catalytic, and it favors the R state, not the T state, which means it favors the enzyme being active. Does that help? Yes? The cutoffs? You mean where the material is? Sure. Okay, so going back, it's actually labeled in the highlights, but I'll show you. If you go back to through the highlights of this, wait a minute, hold on. Enzymes. Yeah, okay, I did have to change the highlights. So this is the end of the material for the first exam right here. Okay? Oh, wait, hold on. What did I do? Oh, you know, I had a problem with something this morning, and I did make a change. So anyway, the new material starts right here. So for the second exam, the new material starts with this set of highlights. That much is for sure. And it ends with the material right at the end of this lecture. Well, actually, it's actually at the end of there. So at the end of transcription, that's where the material stops. Okay? Yeah, sorry for that confusion. I discovered I had two things that were incompatible in the earlier thing about where it started. So, yeah. Yes, ma'am.
Can you go over the release of the enzyme, like when you do kind of
Oh, yeah, the mechanism. Sure, yeah, okay. So how does a serine protease work? How does a serine protease work? Okay. Membranes, nucleic acids. Oh, sorry, it's back here. Okay, so serine protease. Ba-ba-bam-bam. All right, this isn't what I wanted to show you. What I want to show you is here. Okay. No? Ah! Come on, Ahern. Here. Okay. Now, a lot of material here. All right. Big picture. Big picture, we're understanding how chymotrypsin works. Chymotrypsin is an enzyme that breaks peptide bonds, and we're seeing how the peptide bond gets broken. It gets broken in two phases. Okay? First phase is a rapid step. The second phase is a slow step. All right? Yes?
Why is it a rapid step when it obviously is one step and this last one is take on?
It's not the number of steps, it's actually the way each step has to occur. So it's not the number of things that are happening in there that makes it fast or slow. Okay? But there's actually an activation that has to happen. And that second step, you have to get water into the active site, and that takes a while. Yeah. Okay. So, a fast step and a slow step, right? The fast step occurs when the peptide bond gets broken. The slow step happens when the peptide gets released from the enzyme. Okay? How does the peptide bond get broken? Got a catalytic triad. Aspartic acid, histidine, and serine. The catalytic triad is set up to make an alkoxide ion out of the serine. We don't even have to worry about how it happens, but it just makes an alkoxide ion out of the serine. It's the alkoxide ion that attacks the peptide bond that breaks it. That's the fast step. Half the protein gets released at that point. The other half gets stuck on the serine. The second half of the process involves the release of that other half of the peptide from the serine. And that's the slow step. Yes?
What stuff, because the catalytic triad makes an alkoxide ion on the serine, but what got stuck on it?
So, we've got a poly, it's a good question. We've got a polypeptide that's been bound by the chymotrypsin. The chymotrypsin breaks a peptide bond. Now, if nothing else happened, those two pieces would just go flying out and be split into two pieces, right? But the mechanism of the splitting means that one part gets released, but one part, the other part, gets stuck to the serine. So instead of letting two pieces go, one piece gets let go, the other piece gets stuck. The other piece has got to be released, and that's the piece that you're asking about. Okay? Yes, ma'am. Exonucleases on a DNA polymerase, certainly. Yep. Okay. DNA polymerase. Yep, I did it again. All right, if we look at DNA polymerases, those are eukaryotic. There's what we've talked about. All right, the DNA polymerases of E. coli. So there are three. Two we didn't really talk about. 1 and 3 were the main ones. And the one I talked the most about was actually number 1, even though it's not the most important polymerase in terms of making the most DNA. 3 makes the most DNA. But polymerase 1 I talked about because it has 3 activities, and those activities are interesting. It has polymerization in the 5' to 3' direction. That's an activity that makes phosphodiester bonds. That's what makes the backbone of a DNA. The second activity of polymerase I is an exonuclease. Now, to remind you what an exonuclease is, it's something that grabs the end of a nucleic acid and it starts breaking phosphodiester bonds one at a time, chewing in from the outside to the inside. Okay? A 3' to 5', I like to think about it this way. Here's polymerase going along, 5' to 3', 5' to 3', 5' to 3', 5' to 3', mistake. Stop. How do you remove the mistake? You've got to go backwards. 3' to 5', 3' to 5', 3' to 5'. Remove, and then replace. 5' to 3', 5' to 3', 5' to 3'. So the 3' to 5' exonuclease is removing mistakes. It's the proofreader. The other activity is a 5' to 3' exonuclease. And this is the one that's used to remove an RNA primer. Remember, we've got a whole series of Okazaki fragments that are sitting here. Polymerase I grabs a hold of the 3' end of an Okazaki fragment, and it starts making DNA. It's moving now in the 5' to 3' direction, and all of a sudden it hits a primer. And it hits the primer at the primer's 5' end. What does it do? Chew out RNA base, put in DNA base. Chew out RNA base, put in DNA base. Chew out RNA base, put in DNA base. And you see I'm moving in the 5' to 3' direction. That chewing out part is the exonuclease, and it's working in the 5' to 3' direction. Does that help? I forget who asked me the question. Yeah, okay. Yes, ma'am.
I have a question about the mute LHS.
The what?
The mute LHS.
Yes, mute LHS,
In the highlights, you talk about them separately. Do they all do the same thing? Or do
Yeah, I apologize for the thing in the highlights. I grabbed an earlier highlight. In this class, I've only talked about the mute LHS as all basically doing the same thing. So you don't need to know the individual functions of L and H and S. Yeah. Yes? Yeah, what makes a strong promoter or a weak promoter? Okay? It's actually fairly simple in the case of E. coli. It really depends on how well the sigma factor likes it. So if the sigma factor likes it, it'll bind and the polymerase will start transcribing. And if the sigma factor doesn't like it, it won't bind as effectively, and therefore you won't get as much transcription. Now, how does it decide if it likes it or it doesn't like it? And the answer is, how close is it to that ideal sequence that you saw on the screen? That's really what it comes down to. So the closer it is to that ideal sequence, the more the sigma factor will like it. And the farther away it is from that sequence, the less the sigma factor will like it. Be perfect. A perfect TATAAT. Yeah. Yes, ma'am. Okay. So our question was, why is it that we lose a sequence at the end of the DNA of eukaryotic cells? Why is it that that end is not replicated properly? Okay. To answer your question, here is a representation of it. Okay. So if I have a chromosome, it's got 2 strands, right? So I've got one strand that's going 5' to 3' this way, and I got another strand that's going 3' to 5' this way, right? So here's my thing at the end of a chromosome. If I were to replicate, let's say, this bottom strand, the bottom strand, I would replicate, and the 3' end would go all the way to the end, right? Because that's where the 3' and the 5' will be paired with each other, and it would go all the way to the end of this 5' end of the strand, right? You with me? Okay, well, all right, let's see. Let me see if I can draw it. Let me draw it for you. Okay. Somebody want to hand me a piece of paper? I'll draw this out for you. And I guess I need to borrow a pencil, too, since I didn't bring my pen. Here's a pencil. Yeah. Well, maybe I better borrow one. That's not very good. Okay. There we go. Thank you. All right. Let's imagine, if you will, that I have a double-stranded linear chromosome. Oh, sorry. The orientation lights on here aren't right. It looks like, okay, that's fine. All right, so there's a double-stranded linear chromosome. Top strand goes 5' to 3'. The bottom strand goes 3' to 5' because they're anti-parallel, right? If I were to replicate, let's say I peel the strands apart. So I've got a strand here at the bottom, which is the 3' to 5', and I have a strand at the top up here, okay? Let's say I were to start synthesis of a strand, this bottom strand, from here, and I moved it this direction. It would go all the way to the end. How would I start this strand over here? That's the question. Well, it turns out I would start it with a primase. Primase puts an RNA down, and then a DNA polymerase comes along and elongates it. So now, oh, I've replicated the whole chromosome, that whole strand, except for this piece right here. Right? So our cells have something like the 5' to 3' exo that E. coli cells have. So they come along and they will remove that RNA piece. In other words, this piece that was here is now gone. With me? How do I fill this in? The only way I could fill it in would be to start with another RNA primase, because that's how I have to start my strands. I can't start with a DNA. Does that make sense? So because I can't start, I'm always going to have a gap. Always going to have a gap. Okay, now the lagging strand is a different phenomenon, so I'll come back to that. Let me do one at a time. Let me answer this first. So do you understand why there's a gap here, and that gap is never going to be filled in? Okay? And when I go to replicate the top strand, it's only going to get as far as that before it runs out of steam. So every time I replicate, the strands get shorter and shorter. Now, your question about the lagging strand was this, all right? Let's imagine I've got a replication fork, and I'm going, in this case, that's my orientations. The leading strand, I get my RNA primer. I start making DNA. And I'm going in the 5' to 3' direction. Right? At the same replication fork, things can only go in the 5' to 3' direction, but that direction is this way. Okay? Or you say, well, okay, so it just goes this way. My question is, how do I replicate this? I replicate this only as that region gets opened up, and when it gets opened up, another primase comes along and replicates it. And then when this region opens up, another primase comes in and replicates it. Nope, nope. You're not losing these. Well, no. The reason you're not is, remember, we're in a prokaryotic cell here. It's circular. So it comes all the way around. Nothing is lost. Only when we have the linear case is something lost. You got it? Good. Okay, good. It's something that takes a while to get on top of. Other questions about that? It's a common question. Question? Oh, you're just waving at me. Okay. Yes? To show you what? The loop? Oh, okay. Well, the loop is actually, I drew it as a Y, but the loop is like that. Okay? That's the loop. Are you talking about, oh, I'm sorry, you're talking about the big chromosomal loop, this thing, right? Okay. So that's what it would look like. So that'd be a giant bacterial chromosome right there. Yeah, that's a very good question. How does the timing work? Because it's got to do lagging strand, it's got to do leading strand, et cetera. I posted a video of DNA replication. Have you had a chance to look at that video? Okay, let me see if I can pull it up real quickly. Because the video is phenomenal. It's absolutely phenomenal about what all is going on in this process. All right? Let's see if I can get it for you. Oh, blast it. I think that was the one I wanted, but hold on. Yeah, that was the one. I didn't realize that was gone. Yeah, that's just showing it in simple terms. No, here you go. This is it. Okay. This is actually that replication fork. Now, it's a little hard even for me, and I understand what's going on here, to recognize what's going on, but you see that there's a looping out and you see a letting go of that strand. What's happening is that orchestration that you asked about, flipping between leading and lagging, causes this. That's the replication fork right there in the front of you with the blue. It's flipping between leading and lagging. Leading goes faster than lagging. And so what happens is you form a trombone-like, what they call a trombone structure, and that's that loop that's out there. And then when it gets caught up, it lets go of the trombone structure. I didn't talk about the trombone structure because you didn't have to know that, but that's what's actually happening in that process. This is going on at 1,000 nucleotides a second. Pretty incredible. Now, I won't claim that that video answers the question that you asked me, okay? Because, as I said, I look at that video and it's hard for me to orient myself with what's going on that thing. But the point is this. Leading and lagging strand, everything has to be orchestrated. If the cell gets too far ahead of one or too behind the other, then you could imagine you could make some giant knot-like structures and so forth that will happen. Suffice it to say that the proteins that do this have evolved the ability to manage that process. And problems can arise in replication, but we know them as cells that we never see because they die in the process. All right? But that process works, and it works amazingly well at 1,000 nucleotides a second. Yeah. Other questions? Yes, I'm sorry. The DNA ligase is any place where there's two pieces of DNA to be put together. So it's mostly on the lagging strand, yes. Okay? Mostly on the lagging strand. Yeah, go ahead. So the ligase is sealing individual fragments of DNA. So ultimately, it's going to link together the Okazaki fragments that are in the lagging strand. Okay? In the leading strand, when it goes all the way around, it's going to have one place where it's going to link that. Right? Okay. So I said it's mostly in the lagging strand, but there's one place where it will do it in the leading strand. Okay? Yes, ma'am. What are what? What are waxes? Ugly things in your ears? Wax is all I said was it was just an example of a lipid. Waxes are very nonpolar, like many lipids are, and I pointed out that it was I think it's an ester between a long chain alcohol and a long chain fatty acid. Yeah. Yes, ma'am. Yeah. So supercoiling in very simple terms is a structure in DNA, both, any structure in DNA, where you've got something besides 10.5 base pairs per turn. You've got too few or you've got too many. That creates tension in the DNA, and that gives rise to what we call supercoils, which are those things that I showed you on the screen. Okay? And I used the analogy of a rubber band that you coiled up. That's really the best analogy I can give you for a supercoil. And the reason they're important is that, well, there's a complicated answer and there's a simple answer. The simple answer is that supercoiling can cause a problem in replication if it's not taken care of, and that's why gyrase is important. The complicated answer is that supercoiling can affect transcription, and that's an entire course worth of material. The simple answer is that, no, that's fine. The simple answer is that it can affect, if it's not dealt with properly, it will impede DNA replication. And that's why the gyrase was important. Yes, ma'am. Prostaglandins, yes. Prostaglandins are molecules that have a variety of functions in the body. One of them is that they cause pain. And so I talked about how pain relievers work, and they work by inhibiting the conversion of arachidonic acid, which is the precursor, into prostaglandins. If prostaglandins get made, then you will feel pain. And COX inhibitors are the enzymes that inhibit those. And a good example of a COX inhibitor is aspirin. Ibuprofen is a COX inhibitor. The COX inhibitors are also called NSAIDs, N-S-A-I-D-S, non-steroidal anti-inflammatory drugs. Yes. So her question was, isn't the enzyme also called PGH something? It's called, yes, it's called PGH synthase. It's also called prostaglandin synthase, so it has several names, but they all do the same thing. They all catalyze the formation of arachidonic acid into prostaglandins. So the question was, if you inhibit the production of prostaglandins, are there negative effects? And there are. So I've actually talked about a couple of them. One of them was that some prostaglandins are necessary for regeneration of intestinal tissue. So people who take a lot of aspirin sometimes have intestinal issues. There are effects on bleeding. So prostaglandins are necessary for making thromboxanes, which are necessary for platelet stickiness. Yes, ma'am. Sure. So the question was, how can I say again how the prostaglandins are connected to thromboxanes? So Prostaglandins are simply the precursors of thromboxane. That's all I've said about the connection between the two. But the thromboxanes are necessary for making your platelets be sticky. And that stickiness is a factor in blood clotting. So if you don't have sticky enough platelets, then you may have issues with bleeding. And so that's why before you go into a surgery, what in the world's going on there? Before you go into a surgery, they will ask you if you're on aspirin or not, because they don't want you to bleed and have too much bleeding, basically. Yes, ma'am. If you had thin blood, would you what? Vitamin A? K. Oh. Generally, you wouldn't take vitamin K. No. Vitamin K is something that is a pro-clotter, but it's something that would have to be administered in a very properly done condition. You would not normally take vitamin K. You would normally get enough vitamin K in your diet, okay? But the problem with vitamin K is because it's favoring clotting, you really want to be careful not to be favoring clotting in places you don't want it clotting. So, no. That would be under a doctor's control. Yes, ma'am? Yep. Yep. Okay. So our question had to do with the conversion of a double-stranded DNA into single-stranded DNA. And this was a process I called denaturation. Like we can denature a protein, which means unfolding it. We can also denature a DNA molecule, and that simply means pulling the strands apart. We can pull the strands apart in several ways, but the easiest way is to simply heat it up. Okay? As we heat up a duplex DNA and we monitor the absorbance of light that DNA molecule has, we discover that as we heat it more and more, the absorbance goes up. And the reason it goes up is because the single-stranded DNA absorbs light more effectively than the double-stranded DNA did. Okay? And the wavelength at which that occurs is what you said, which is 260 nanometers. But, yeah. You don't need to know the 260 nanometers. You got that look like, God, he's got one more thing he's giving us. Yes, ma'am. Linoleic and linolenic? Sure. So there are different fatty acids that I talked about in class. And I talked about saturated and I talked about unsaturated fatty acids. And I also talked about polyunsaturated fatty acids and polyunsaturated fats. By the way, don't confuse fatty acids with fats. I find that that's a common problem that students have, is they confuse a fatty acid with a fat. So a fat is when you combine 3 fatty acids with a glycerol. That's how you make a fat. But a fatty acid is not the same thing as a fat. Okay. Now, your question had to do with linoleic versus linolenic acid. Okay. These are both fatty acids that are unsaturated. Linoleic acid has 2 double bonds. Linolenic acid has 3 double bonds. And I'm going to give you the numbers, but you don't need to know the numbers. Okay? Linoleic acid has double bonds between carbons 9 and 10 and between 12 and 13. Linolenic has them between 9 and 10, between 12 and 13, and between 15 and 16. Now, the only reason I mentioned those numbers to you, you don't need to know the numbers, is because mammals can't make fatty acids beyond position 9. So we can't make linoleic, and we can't make linolenic, which is why we call them essential fatty acids, because we have to have them in our diet. Does that help? Okay. I always forget who asked me the question. Somebody asked. Somebody over here, I remember that. Okay. Yes, ma'am. On what? Nicotinamide? So the NAD? Okay, sure. So nicotinamide is it's actually a nicotinamide adenine dinucleotide, which is what NAD stands for. Nicotinamide is a part of that. NAD is an example of something I called a coenzyme. So a coenzyme is something that is a non-amino acid that helps an enzyme to do its thing. In the case of NAD, it can accept electrons and protons that are generated by oxidation. Yes, ma'am. Well, it doesn't do it to keep it neutral because it's actually going from NAD plus to NAD zero. But if it goes the other direction, it goes from NAD zero to NAD plus. So it's not staying neutral. But chemically, it's easier for it to be stable with that proton that's there and not just accepting the 2 electrons. Yeah. But cold. Cold. You know, I called earlier in the term, and I had them turn the temperature up in this room. But today, for some reason, it seems like it's cool. It was cold earlier in the term, but I think they've gotten better about it. So think warm thoughts. Yes, ma'am. Okay, the tryptophan operon. So the tryptophan operon is an operon. So an operon is something that's a collection of genes in prokaryotes that's under the control of a single promoter. That's what an operon definition is. A collection of genes in prokaryotes under the control of a single promoter. The tryptophan operon specifically has 10 genes in it, and those genes are necessary to code for proteins that are involved in making tryptophan. Necessary to code for proteins that are necessary for the cell to make tryptophan. When the cell has little tryptophan, it wants to make tryptophan. When the cell has a lot of tryptophan, it doesn't want to make tryptophan. The operon is set up so that when the tryptophan concentration is high, transcription starts but gets terminated early. When tryptophan concentration is low, transcription starts but doesn't end early. It continues all the way through all 10 genes. And that's the difference between the two. Hopefully this will be the most that you'll have to learn for any exam. Anything else? Okay. I will be in my office tomorrow. If you have questions, come see me.